Hi everyone, welcome. Today we're joined by Dr. Stephen Schmidt and we will be discussing the microbiome. I guess one of the unknown and exciting frontiers to solve many of the problems that we are facing in modern day medicine. Stephen, welcome. We're looking forward to what you have to say.
Thank you, young man, and hello to everyone, and thank you for that introduction. Yes, okay, so we are going to be talking microbiome, and I'm not quite sure that it's going to be weekly. It's going to depend on how hard we work and how quickly we can get these things together. But we will be doing the full thing on everything that we know in a 5-part series that I sort of worked out myself. Okay, so I am sort of 3/4 way through my life if I reach the predicted age, and I'm actually sorry that I'm not 30 years old, because I strongly believe that this concept of genetic interaction between bacteria and fungi and archaea and our bodies is the future of medicine. I truly believe that. And I'm gonna try and demonstrate to you why I say that. Every practitioner, every health person, and every individual that's concerned about his health should think about his microbiome. And that statement is a fair statement. And in the next 5 or 6, 8 maybe weeks that we're going to do this, I'm going to try and explain why I say that with some cases in the end. And Anton and Stefan, Professor Colleen is going to help us to cover this whole area from the technical stuff through to the clinical applications. The first thing that we have to understand is, that I want to say that is not depicted on the slides, is that the areas of the body that is inhabited by microbacteria and microorganisms is all the areas that communicates with the outside world. So you have your upper respiratory tract, your sinuses, you have your skin, you have gynecological system, and of course the gut from mouth to rectum. So it is really the communication between the outside world and your organs. And there's an interface there which I will explain. This little picture here is done on the inside. It's a graphic. It's not a very good graphic because the colon doesn't look like this. This is a small bowel with some bugs in it, but just to give the idea. All right, so let's go through some basic things. And I'm planning to do with Anton and the others, we're planning to do this in more or less this format. All of this will make much more sense later on. So basic information, things we call diversity, structure and function, commensals, keystone bacteria and pathogens, clinical and therapeutic applications, products, and gut mapping. All right, so the number of microbes differs from article to article. The most recent research says there's about an equal amount of microbes to human cells, but this specific article calls it 100 trillion microbes. I don't think it really matters. It's a hell of a lot. The microbiome is called the hidden organ because it functions as a full organ. When you take the genome, the human genome, our genes, and you combine that to the microbiota, which are the organisms, that is called the microbiome. So the bugs are the microbiota, and the microbiome is the gene collection. All our metabolic features is a combination of human and microbial genetic effect. And what has brought to us in the last, I don't really know how long, last, say, decade and a half, maybe 2 decades, to this point is to identify bacteria and microbes. Previous technologies had to depend on culturing. And the culturing is obviously a laborious task and a difficult task in many cases. I mean, you take Helicobacter pylori, for instance, you battle your backside off to get Helicobacter cultured. So I think the figure is about 1,000 organisms have been they've been able to culture mechanisms. But now what we're doing is we're doing sequencing and genetic identification of bacteria. So just some definitions to quickly go through. Microbiota, as I said, are the actual organisms. The biome are the genes of the organisms and the gut. Eubiosis is a good balanced microbiome. Obviously, dysbiosis is an unbalanced microbiome with symptoms. Diversity is a very important thing. That is basically your rainbow picture in color of the wide array of your microbes. Prebiotics is basically your diet and other things that you can take in that would stimulate the growth of microbes. Sporebiotics, I haven't given a definition. It's Anton Janssen Ferensberg's baby, and he's going to tell us a lot about sporebiotics, and it's a very important thing. But basically, you can look at sporebiotics like little infants of bacteria. So it's the baby bacteria that can grow into something else. Probiotics, we all know, those are the bacteria itself. And symbiotic is a term in research. If you read an article, they talk about symbiotics. It's basically a combination of your prebiotics, which is the food, and probiotics. We simply, according to most articles, know between 5 and 10% of what is going on in the microbiome. The majority of it simply don't know. Bacteria are very well defined. The viruses and the fungi and the archaea is not that well defined. So the makeup of the gut microbiome is bacteria, fungi, viruses, and archaea. Archaea are prokaryotic cells without nuclei, previously also called bacteria, but has since been differentiated. This is important, even though it looks very boring, because if you read these things and you read all these horrible, unpronounceable names, you basically got to always look. You can always Google this very quickly. Wikipedia is very good at this, to tell you exactly where things lie. So the kingdom is the larger group, as in bacteria. Phylum is a family description. Class is not all that important for us. The genera is sub-classification, and the species. And if you had to go and look at something like Lactobacillus reuteri, which is a well-known probiotic, it would classify as a bacteria in the kingdom. It would classify as a firmicute in the phylum. Then the class is Bacilli, the genera is Lactobacillus, and the species is Lactobacillus reuteri. It's important for the interpreter of a microbiome analysis to know the stuff. And I think it's important, especially the class and the species, for everyone to know when you are going to manage someone with a probiotic or a sporebiotic or symbiotic. This is a beautiful little graphic that I stole from the internet that actually explains to us what exactly is going on here. What are we looking for? So amplicon sequencing and shotgun sequencing are two technical terms for how they get to the actual name of the bug. Amplicon sequencing can tell you about the structure, and I'll just tell a little bit more about that, and the interaction between the bacteria. Shotgun sequencing can tell you about function and interaction, and metagenomics will collect all of this together. I'll explain it to you in the next slide and make it a bit simpler. So amplicon sequencing, or just finding the structure or a differential analysis, is basically saying to us, what is there? What are the bugs? What are the names in this collection of microbes? The metabolic pathways or the metagenomics will tell you, what do these bugs do? That's a vast field that is just scratched on the surface at the moment. So there are some of these very expensive analyses which can actually tell you this bug does this and this bug does that. So that is the function of those microbiota. And then a very, very important thing are the keystone bacteria. And I'm going to try and explain to you with a simple analogy. Pardon me if it sounds too simplistic or condescending. So let's say that this random group of people that I got from the internet is your total microbiota. And I'd say to you that a portion of them are English-only speakers, and a portion of them are Afrikaans-only speakers, and a smaller portion of them are bilingual. Those would constitute your phyla, or the big family, just under the kingdom. And we have 4 or 5 very important phyla in our bodies. I'll go a little bit further on that. But out of this group, you can then group them into phyla A, English speakers, in the microbiome, that would be the firmicutes. Phyla B would be the Bacteroidetes, and then you've got Actinobacter, and you've got Proteobacter and Fusobacterium. Now, here's a very, very important thing I'm going to show you now, and this is why I use this analogy. If you look at these 4 gentlemen, they could be classified as language teachers. They could be either English or Afrikaans or bilingual speakers. In other words, they can be from any of the phyla, but what they are is they are language teachers. They are the keystones. So this guy can all of a sudden go away, and he was the English teacher, and then you'll find fewer English people in future, if you understand what I'm saying. So what they go across phyla, and they organize the larger groups. So if you look at what you find when you do an analysis, this is actually comes from my gut, sorry for that, but this is a bacterial species. All the species that were looked for listed in percentage representation. And if you look at a report, a good report, then you will find this little rainbow bar, and this is diversity. If we are going to do gut analysis, and you want the very first thing you want to know is, how diverse is my microbiome? There is an index called the Shannon index, which gives you a number. It's either 1, 2, 3, 4, 5, 6 or more. I'm still trying to work out exactly how that works. It's a rather complicated formula. So diversity is the #1 thing for your microbiome. These are the phyla, or the big families. Of all the names you've got to learn, you basically have to know what the firmicutes, Bacteroidetes, Actinobacteria, and the Proteobacteria are. So these 2 are the largest groups. They make up the majority of the microbiome, more or less in a ratio of 2 to 1. So 90% of the microbiome will be sorted into 2 phyla, or 2 large families, and the ratio is more or less 2 to 1. They do different functions, and we'll get to that later on. We can talk about that as well. Actinobacteria contains many of the keystone bacteria, and Bifidobacteria falls into this little group, the Actinobacteria, which makes up about 0.2% of your gut. The Proteobacteria are usually the bad ones, and Fusobacteria is not important for now. Okay, here's a depiction of the concept of the keystone bacteria. And this is a large article that I read, very complicated article, but this was the picture that came from it. And if you look at this bar here, this is a healthy person. Each of these groups would constitute a bacterial group. And you can see there is a keystone bacterium. And that keystone bacterium, why it is called a keystone bacterium? Because it regulates most of the others. So it is the language teacher that can either create more of these language speakers or fewer. In the diseased person, if you compare those 2, the keystone is gone, which has led to the overgrowth of the orange bar. I think I've put that clearly. I hope you get that. So where does the word come from? This is just an interesting little observation, or where it comes from that I looked at. This is a study done by a zoologist called Robert T. Payne. And what he showed was, in a tidal pool, you have many species of sea living creatures. The purple star is the major tidal pool predator. So what he did was he removed the purple star from tidal pools. Before the removal of the starfish, these were all the critters in this tidal pool. Then he removed them, and after a year, that was what was left. And after 5 years, he only had mussels. Mosselby. So you see these tidal pools here where I live, it's just mussels all over the show. And that is basically the loss of your keystone predator in these tidal pools. And that is an extremely important part that you have to understand if you're going to understand the microbiome and if you're going to manage people with dysbiosis. So basically, the tidal pool goes from a very varied and a diverse group of sea creatures to just a gewone ou mosselbaai poeltjie met baie mossels. The keystone bacteria are listed here. I'm not going to go through them, but you'll see the bifidobacteria is very prominent there. And the definition here, the keystone taxa are highly connected taxa that individually or in a guild or a group exert a considerable influence on the microbiome structure and functioning, irrespective of their abundance across space and time. These taxa have a unique and crucial role in microbial communities, and their removal can cause a dramatic shift in microbiome structure and functioning. And I'm going to say a little thing right at the end. Remember that little statement. Their removal will cause chaos in the gut. So where did these things come from? I don't want to go through this in detail. I just want to mention one or two things. So it takes about 3 years for the microbiome to be settled well, it was said previously from birth, but it's actually, we know now it's already in pregnancy. So it's about 3 years that it's settled. An important thing that I found in an article was that, okay, let's just go through this. In pregnancy, it's placenta, meconium, amniotic fluid that feeds microbiome to the fetus. Birth is obviously the birth canal, and there's also the skin canal with a cesarean section. And this is very important. Lots of moms have been vilified and labeled that they are a cause for their kids' abnormal microbiomes. But it's been shown that between normal delivery and C-section, the delivery equalizes after 3 years and is similar to the mother's microbiome. It takes a while longer, but it gets there. An interesting thing that I read, and I've lost the article, so I cannot quote it to you now, I'm going to find it again, is that there are many people, I think this comes from the United States, where they're actually doing a vaginal microbial transplantation for C-section babies by smearing vaginal fluids onto the fetus after birth. And then breastfeeding, first 3 years, nutrition, exposure, pets, and of course all the stuff that can destroy the microbiome after that. I've already said that these are the groups where the microbiome is present. We're going to concern ourselves with the gut. Okay, so what does it do? If you can remember this picture in your mind, you're going to understand the microbiome. Here is the lumen of the colon, and this is where all this is the electron microscopy photo. All right, so here are the bugs. This green thing here is the mucus barrier, and the blue dots over there is the colon and lymphoid tissue. Right, so if you that is just a graphic explanation of it. And bugs, mucus layer, colon cells. Now, what is very important here, there are many things that the microbiome interacts with and with the body. But two massive things that happen here is that a lot of these bacteria in certain phyla and species actually regenerates this mucus layer to keep it nice and thick. Overgrowth or dysbiosis of these bugs of certain bacteria are mucolytic and breaks down this mucus layer. That's very, very important. Something that I learned not long ago, which I didn't know, was especially the thermosuits produces short-chain fatty acids, and specifically butyrate. And butyrate that is produced here feeds the colon epithelial cells directly. So if you have a lack of butyrate producing bacteria, your colon cells are under stress. One of the not the biggest causes, but a very prominent cause in constipation, because you lose peristaltic activity. And then, of course, the barrier must be a good barrier, because I want to show you just now, the gut microbiome from birth, actually before birth, from pregnancy, has positive effects in developmental brain development and behavior, protective effects, immune stimulatory effects, and basic role in just breaking down stuff, vitamins, production of vitamin K2, utilization of B12, feeding the colon. And of course, when things go bad, then you get diseases. This is a very complex slide, but it's actually very beautifully worked out. But I just want to highlight this part here. If you can just concentrate on this and see that the one problem that we have with interpreting the gut is the fact that the brain will control the gut in a downward control, and the gut will control the organs, and especially the brain, in an upward control. This article comes from a 46-page article that deals with microbiome and neurodegenerative disease. So the way that the gut and the brain communicates is through cytokines and toxins that go through a damaged barrier, directly through the vagus nerve, sorry for the spelling, and the enteric nerves. It cruises up in the nerves, and hormonal immune cells and neurotransmitters. I've halved that complicated picture for you just to look at a few things here. So this is a healthy gut. That is just to show that this part is the lumen and that part is the wall. You will notice the bacteria in the mucus layer. You will notice a healthy thickness of the mucus layer, and you will notice that the cells are tightly junctioned, like my fingers, so the junctions are tight. On the other side of this fence lies all the good things that has to happen, or the potential bad things that has to happen, the lymphoid tissue, the hormonal control. I'm not going to go into that now. It becomes very complicated. But basically, in this gut, you have tight junctions, you have well-fed epithelial cells, and you have the immune system is under control, and you don't have any abnormal events happening. Good fences make good neighbors. If things are right there, all of these things in place, everything is hunky-dory. Now, on this right-hand picture, you have got a thinning of the mucus layer. It says an aged gut, because it refers to the neurodegenerative stuff, but a sick gut would do the same. Right, so now what you have now is you have separation of the cells, and you get leakage, the so-called leaky gut. So bugs come out here, lipoproteins, polysaccharides come out, toxins come out here, and now there is an intrusion of everything that is happening down here. Now you have the potential for increased leakage of bacteria, bacterial metabolites, LPS, toxins, DNA, mucus, all the junk that the colon can leak through that can affect the immediate immune system. It can affect the colon cells itself, go into the portal system and affect the liver, go up the enteric plexuses and the vagus plexus and affect the brain. So you can add to this list, but these diseases that are listed here actually have decent research, reproducible research, to show that dysbiosis either causes or exacerbates or contributes to all these illnesses. And pretty much you all know this describes all chronic illness in a bubble. So this is how I see it. This is my opinion, might not be completely correct, but to go from a healthy person to chronic disease, you basically have genes that could be a problem, and we know that's about 5 to 10%. All the environmental things that go wrong, which you've spoken about ad nauseum, and it works through a system of inflammation, oxidation, toxification, mineral depletion, and biopathway interruption. So these poisons are causing disease by these metabolic processes. And I actually believe that the microbiome is the gateway between those two. Because if you learn and you read about how these bugs in the colon actually causes the disease, it goes through these kinds of processes. Inflammation, oxidation, toxification, mineral depletion. For instance, if you get overgrowth in your small bowel, you get bacteria that compete for vitamin B12, and you cannot absorb your own B12 that you eat. So the microbiome and dysbiosis goes through the vagus and the enteric nervous system to chronic disease, toxin release through a leaky gut, immune dysregulation, and malnutrition, or poor absorption of nutrients. That's how it gets to chronic disease. And then through the hypothalamus, pituitary, adrenal axis, which is a very, very complicated thing, but it's quite astounding to see how that actually works. So I'm almost finished with tonight. So just a few things that is fascinating from the research and is cast in stone. And this is a very landmark, a fantastic study and a landmark study that not started the ball rolling, but started people believing in the actual effects. So the top 3 little critters here, with the stop signs here, means that they are completely sterile. Their guts have been made sterile, okay? In the left-hand mouse, what they've done is they've donated the microbiota of a normal mouse into the germ-free mouse, and the phenotype of the mouse and its offspring would be normal weighted. Then they take an obese interim, stick it into the germ-free mouse, and you have an obese mouse. And the same with anorexic mice. So what is happening here is you have a perfect model of a sterile or a germ-free mouse, put in the phenotype of weight, looking at weight differences into the germ-free mouse, and you get different body makeup. Okay, so if there are any questions, please shoot. We're still going to do everything in detail. There are incredible things to show you and to demonstrate from research and from articles. Almost every one of these chronic diseases has a little recognizable package of bacteria that are either in abundance or depleted. So we're going to be able to sort of identify groups of bacteria in disease. And the next thing we would like to do is then, if we can do gut analysis, is to say, okay, now we can target specific problems.
Yeah, okay, thanks, Stephen. So appreciate what learning from you. We are all learning. Yeah, thanks so much. Until we meet again to discuss the microbiome.